Hello, and welcome to a short video that demonstrates and explains the action of the lac operon found in E. coli. E. coli is a common bacteria. It inhabits our guts. It's very useful for us. Sometimes it can cause disease. But the lac operon is a very good model of how genes can be regulated. There are many different ways that genes can be regulated, turned on or off, but the lac operon is one of the best studied. And for AP biology, this is the one that you definitely should know. Okay, so we're going to start this, and starring E. coli here. This represents the lac operon region. That is a short piece of DNA within the bacterial chromosome. So what we have here are different regions of the gene. The lac I, the promoter, the operator region, and then three regions, lac Z, lac Y, and lac A. The whole purpose for this gene is to produce an enzyme called beta-galactosidase that will break down the sugar lactose. So lactose is a sugar commonly found in dairy products. Some people cannot digest it. They're called lactose intolerant, but this is a different situation. The reason to have gene regulation is that it takes a lot of energy to transcribe to messenger RNA and then to translate that messenger RNA to make proteins. It takes a lot of energy, a lot of ATP. The cell doesn't need to turn on this gene if there's no lactose present. So the lactose went away here. Okay, this represents the lactose, a little green, and it's not there. Okay, so it's not in the cell, and no beta-galactosidase enzyme is being made. So ignore this portion down here. Only concentrate on this. The lac I gene here encodes for a repressor protein. The repressor protein is a protein that binds to the operator region here and blocks RNA polymerase from transcribing the gene. So when the gene is on, it's not on now, but when it's on, it transcribes these genes here. So let's review a little bit here. Okay, so lactose, again, is an enzyme that breaks down lactose to glucose and galactose. Those sugars can now enter into cell respiration. But if there's no lactose present, so the lactose went away here, there's no reason to turn on the gene. So the lac I gene encodes a repressor protein shown in red here. Here's the messenger RNA from the lac I gene and the repressor protein binding to the operator. So repressor protein binds to the lac operon in the operator region, and it blocks RNA polymerase. RNA polymerase can't get past here and cannot transcribe the gene. So the gene is now off. It's not making any messenger RNA. No protein is being made. It's turned off. It's saving the cell energy. After all, there's no lactose present. But now, lactose is present. Let's say it gets into a glass of milk. So lactose present in milk. So here comes the lactose, shown in green, and that binds to the repressor protein, shown in red, and it changes the shape of the repressor protein. So now the repressor protein cannot bind to the operator. So there it goes away. So the repressor protein falls off the DNA, and RNA polymerase can now transcribe this gene. So here goes the RNA polymerase transcribing the gene and making messenger RNA. Remember, messenger RNA is made 5' to 3'. Let me turn it around here. Okay, so made 5' to 3'. So the RNA polymerase can now go through and polymerize the messenger RNA. And you could have multiple RNA polymerases all following each other. I've only shown one. And now the messenger RNA is made, and it can be translated into these 3 proteins. Now, this RNA, like many RNAs in prokaryotes, not eukaryotes, is called polycistronic. It can encode 3 proteins. Okay, there's a start codon and a stop codon for the LAC-Z, start and stop for the LAC-Y, and start and stop for the LAC-A. So that's called polycistronic. Most eukaryotes do not use polycistronic RNAs, messenger RNAs, but prokaryotes do. Did I say eukaryotes? Eukaryotes don't use polycistronic. Prokaryotes do. So the 3 genes, or the 3 proteins that are made from this, are LAC-Z, that encodes the beta-galactosidase. That is the enzyme that breaks down the lactose. LAC-Y is a permease that lets lactose into the cell, and LAC-A encodes a transacetylase that prepares the lactose to be broken down by the beta-galactosidase enzyme. So now we have the messenger RNA translated into beta-galactosidase, permease, and transacetylase. Here comes the beta-galactosidase, and it breaks down the lactose. So the lactose is gone. Now imagine there are millions and millions of molecules of lactose in there. Okay, but the beta-galactosidase, being an enzyme, can be used over and over again, breaks down all the lactose. And now there's no lactose left in the cell, so the operator is now bound by the repressor protein. So the repressor protein binds the operator region that blocks RNA polymerase from transcribing the gene. So now there's no more transcription and the gene is off. After all, why go to all that trouble and expend all that energy to make enzymes that the cell doesn't need? I hope this has been helpful. You can find this described in the video I attached on Veracross and also in your AP Biology book. Hope you enjoyed this. Thank you.